And then 13 different, in green, 13 different RNA protein complexes. So DNA protein complexes, RNA protein. And their slopes. This we compare here. This is an unpublished work as yet. Compared with experimental, some experiments done. One was published in Science Journal. We compared with their experiments. And one experiment was done in our lab itself, the blue one, in Kolkata. So compare with them experiments, our calculations, and that separates DNA protein and RNA protein. DNA protein in black, RNA protein in distribution, so right. Binding energies, calculated binding, electrostatic power of binding energies, and how. And different many results, maybe may not be much interested, but yes. Yeah, this gives this one, that one. A message to experimentalists, because these are theoretical calculations, computational calculations, theoretical or because electrostatic energy, how will you measure that in laboratory? Difficult to do that, but energy you can measure, you can somehow, but electrostatic part of binding energy. So if we do experiments at different salt concentrations, then we can have a way. With salt concentration, what will vary is the electrostatic part of energy believed. So from our calculation, one can predict. So there one message is there for experimenters. Okay. Another one, molecular dynamics simulation of a peptide. That peptide was a part, peptide as I said, amino acids, chain of amino acids, but short one. Protein is a long chain and then coiled and supercoiled having a three-dimensional structure, globular kind. But one human lab protein, that protein is inside our cells, lab protein. What it does, it helps some foreign RNA, hepatitis C viral RNA, when it infects our cells. Our protein helps that to transcribe and then translate and multiply. The virus multiplies. So what we did, we took that protein in laboratory, excised that part which binds with the foreign RNA, hepatitis C viral RNA. And for that, experimentally it was done by Yasir Khanna. I showed the picture. NMR structure was formed, and I used that structure and also one initial structure from protein data bank of same protein excised from there computationally, and did molecular dynamics simulation of that peptide. 7 were peptides, 7 amino acids: lysine, AYKETDL, lysine, lysine, glucamic acid, threonine, aspartic acid, leucine. Some of them are positively charged at 7 pH, normal pH. Some of the amino acids. This work was published in 2019. One Professor Umakant Tripathi is here at IIT-ISM itself. So he also later on, in later part, he got involved and in spectrochemica at he published. So we are talking about energy. I kept the titles like energy and energetics, but these are the energies at molecular scale that we are talking about. Anyway, when we talk about energies at life size scale as well, in our length scale, so things are taking place at microscopic and nano level. And then so efficient utilization of energy at molecular level inside our cells, in biological systems, in chemical systems, that take place there. So maybe that efficiently we should use energy at our length scales. So always we try to mimic nature. I will not go into details of everything. So that's the peptide. Again here a result is being shown. So 7 were peptide Then these are water molecules, red and white, and they are some salt ions. And we calculated RMSD, root mean square deviation of all amino acids, and the part in that backbone loop of peptide which moved much had bigger RMSD that are more red, and which moved less, deviated less from their equilibrium structure. So the result of calculation is also. And the simulation parameters, configuration files, NAMD software we used, nanoscale molecular dynamics, along with visual molecular dynamics, VMD, and different NMR structure that was found in our lab. RMSD, and again I will not go much into the details of things. It could be a potential drug against hepatitis C virus. Another work, this is simulation of process of target search. What is target search? Protein binds to DNA, as we saw at many places there are DNA protein, RNA protein interactions. So proteins bind to DNA, but DNA are long chains, though coiled and supercoiled. So some lakhs or crores of base units, nucleotide units, monomers. And two double helical structure, two strings are there. So protein binds to some part of DNA, sometimes genes that express into some protein. So, and that part is some 16 or 17 base pair long. So very small length compared to the whole DNA length. So protein binds to that. Proteins binds to that specific site. It will take some time to locate its target. And if it is a normal chemical diffusion process, then it will, whatever time it takes, what happens in vivo, in biology, inside cells, living cells, is it locates its target 1,000 times faster. So, can we explain biology through chemistry? So there, our work was there. So simulation of the process of target search by a repressor protein over a genome, the whole DNA, to explore the presence of some quasi-specific sites. Because they are specific sites, and we found that there are some semi-specific sites. So that is how it locates its target fast, some 1,000 times faster than chemical diffusion process. So that work was published in Biophysical Journal in 2011. In fact, this work found a basis for our another work which we did at our Vinoba Bhave University.